Hello. Can you hear me? Okay, great. Thanks. Just a couple announcements before we get started. The next grand rounds is December 19th. And it will be Danielle Fixon talking about growing old, going green, exploring cannabis in older adults. Very interesting topic. And our next journal club is on the 12th via Zoom, and the presenter will be Elizabeth Bloman. I believe I'm saying that right. I hope I'm saying it right. Okay, how do I say it? Blumen, sorry, Elizabeth Blumen, my bad. Other announcements, Geriatric Division meeting following today's presentation. You'll also receive an email at the end of the presentation with the link to today's presentation and a copy of the PowerPoint and an online evaluation. So if you could complete that, that'd be great. And if you have any questions, please submit them in the chat box, the Q&A box online, and we will get to those towards the end. So without further ado, I'd like to introduce Dr. Zach Clayton, who is talking today on Today's Cancer Patient is Tomorrow's Cardiovascular Patient, getting to the artery of the matter. Zach joined the division in May of 2024 after completing a 4-year postdoc at CU Boulder and 2 years as an assistant research professor at CU Boulder in the Department of Integrative Physiology, which is one of the top integrative physiology departments in the United States. He has a Ph.D. from Oregon and a bachelor's and master's from San Diego State University. His research is translational in nature, and such that he can use cells and preclinical rodent models with a goal of informing future clinical therapeutic strategies in his field of cancer. And the overarching goal of his research is to understand how cancer and chemotherapy treatment influence the cardiovascular system and use this information to inform therapeutic strategies mitigating these effects. So with that, I'd like to hand it over to Zach to talk about today's cancer patients and cardiovascular disease.
Thanks, Josh. You guys hear me okay? Great, great. Awesome. Here we go. Well, thank you, the Division of Geriatric Medicine and the GREC for having me today. Appreciate it. For a brief outline today, just to orient you to the structure of the conversation that we'll be having. First, talk about cardiovascular disease risk and advancing age. And then the influence of arterial aging and how that influences CVD burden with aging, with the goal of setting the stage for cardiovascular aging and how we can use what we know in that space to inform how we go about understanding these mechanisms in cancer survivorship and informing future therapeutic strategies. And then we'll go into cancer risk and advancing age, cancer chemotherapy, and premature arterial aging. So I hope you can now see that connection between how we're using this information up here to inform how we think about these processes, and some of because there are a variety of redundant processes, it allows us to do that. And then lastly, some therapeutic opportunities that I think have started to unfold in our research and in the field in general. So without further ado, here's some data from the American Heart Association showing that cardiovascular disease is by and large the leading cause of death worldwide. And advancing age is the primary non-modifiable risk factor for the development of cardiovascular diseases, as approximately 95% of all cardiovascular diseases occur in middle-aged and older adults. And as I'm sure this entire audience is well aware, the number of older adults in terms of the percentage of the population and the absolute number has steadily been rising decade after decade. It is projected to only increase. So if you take the previous slides mixed with this, if without proper intervention, the healthcare system is going to be met with some significant cardiovascular disease burden, if not met with proper intervention. But why does aging increase cardiovascular disease risk? A key antecedent event to cardiovascular aging is what we call arterial dysfunction. And I'm not the only individual that studies this in our division. So this is a consistent concept throughout our division and the physiology research here in the Division of Geriatrics. But arterial dysfunction we think about as arterial stiffening. So this is stiffening of the large elastic arteries, which is the carotid artery and the aorta. And impaired endothelial function. So that's an inability of a blood vessel to dilate adequately when given a certain stimulus. So just to orient you a little bit more to what I'm talking about in terms of stiffening of the large elastic arteries, we're talking about these two conduit arteries, the common carotid arteries and the aorta. And here's just an oversimplified schematic or a cartoon of what these vessels start to look like with advancing age. So you see this elasticity in a young age, but as we start to age, we get more rigidity to the vessel, which is largely due to collagen deposition and reduced elasticity from elastin. And these elastin properties of the vessel is important to dampen pulsatile flow, or these pressures that are transduced through these vessels. And you can appreciate from this anatomical structure is these conduit arteries are conduits to all of our end organs. So if we don't dampen the pressure adequately, these end organs are being faced and exposed to high levels of pressure, which could be damaging to the fine microvasculature that feed those organs. So what I'm representing here is these higher swings in pulsatile flow. And indeed, large elastic artery stiffening has been independently implicated in overt cardiovascular diseases, Alzheimer's disease and related dementias, impaired vision, and kidney dysfunction, all disease states or conditions that do present with advancing age. And most importantly, as it relates to cardiovascular diseases, large elastic artery stiffening is an independent risk factor for systolic hypertension, which some may refer to as the silent killer. So mitigating systolic blood pressure, perhaps through the lens of large elastic artery stiffening, may be a key way to mitigate CVD burden with this increasing number of older adults in our population. So now to endothelial dysfunction. What we're referring to here is the vascular endothelium, which is a single cell layer lining the inner wall of our arteries. And the endothelium is responsible for producing and releasing the mostly vasoprotective and strongly vasodilatory molecule nitric oxide. So nitric oxide can be released from the vascular endothelium and diffused to the vascular smooth muscle, where it can promote smooth muscle relaxation and vasodilation. But with advancing age, we have reduced nitric oxide bioavailability. And this is an independent risk factor for overt cardiovascular diseases such as atherosclerosis, coronary artery disease, hypertension, and stroke. So this is more of a cartoon schematic of what this might look like. So we have a variety of stimuli that can come in. In humans, we utilize mechanical stimulus such as flow. And in our animal research, we use chemical stimuli such as different drug compounds, both of which can promote nitric oxide release in the vascular endothelium, which is noted here in these tan boxes. But upon this stimulus, this nitric oxide as a gas can freely diffuse to vascular smooth muscle and promote vasodilation and vasorelaxation. But unfortunately, with advancing age, we have reduced nitric oxide mediated endothelial function. So this is a primary risk factor of the development of those overt cardiovascular diseases that I showed in the previous slide. So how do we assess arterial function? And I like to share this to paint the picture for the translational relevance of our human to mouse work that we do. So in humans, aortic pulse wave velocity is considered the reference standard non-invasive measure to measure arterial stiffness. And so what we do is hook a participant up to an EKG so we can get a per cardiac cycle read on the arterial waveforms through the carotid artery and through the femoral artery. And then we can look at the time delay in the given cardiac cycle that it takes the blood to travel from the carotid artery to the femoral artery. So then this is an assay of how quickly blood is traveling through the aorta. And if we take the time delay between the blood traveling through the carotid artery and down to the femoral artery, and then also measure the distance between these two locations, we now have the means to calculate velocity as distance over time. And so if you recall, a stiffer vessel would be reduced elasticity, and there's reduced dampening of that flow. So we actually would see a higher pulse wave velocity if the vessel was stiffer. And in a relatively crude analogy we like to use is a hose left out in the sun for a long period of time. It can't stretch when given a volume or bolus of water, and that's what these vessels start to do as we age, unfortunately. So a higher aortic pulse wave velocity is indicative of a stiffer artery. And relevant to later on when I show you some data, if we can show a reduction in aortic pulse wave velocity, that would be indicative of improved arterial function and presumed reduced cardiovascular disease risk. So in mice, we can't, because the vessels are so small, we can't directly image or look at the carotid artery and the femoral artery in vivo, so while the animal's alive. But what we can do is put the mouse under anesthesia and use a almost identical approach, but this time assay directly the aortic arch to the abdominal aorta and look at the time delay relative to cardiac cycles. So here's an EKG trace. and measure the distance between the two probes. So now again, we have distance over time and the ability to calculate aortic pulse wave velocity. And apologize if you are adverse to mice, but this is what this looks like in mice. So we have mice on a nose cone of anesthesia, and we have two Doppler probes here on the aortic arch and the abdominal aorta, and this is feeding onto a computer screen next to it. And then we have calipers where we can measure the distance. And importantly, what I also like to show this is that we see similar magnitudes of difference if we compare young and older adults and young and old mice relative to the aortic pulse wave velocity measurement. So this then tells us if we didn't see these same similar magnitudes in mice, we could infer that their mice aren't a proper translational model of human cardiovascular aging. But see, since we're seeing similar magnitudes of difference and pretty consistently seeing this, we do believe the old C57 Black 6 mouse is a translational model of cardiovascular aging. So how do we study this endothelial function in people? So this is a technique called brachial artery flow mediated dilation. So we take a baseline ultrasound image of the brachial artery, and then you inflate a blood pressure cuff just distal to the elbow to occlude blood flow. And after 5 minutes, you release the cuff. And then this is that mechanical stimulus that we were talking about earlier when we showed the cartoon schematic of nitric oxide release from the vascular endothelium. And that mechanical stimulus creates a nitric oxide release in the endothelium, and it promotes vasodilation. And you can see this difference after in vessel diameter relative to the baseline. So this difference, the delta, would be considered the flow mediated dilation. And that is largely nitric oxide dependent. And that's in humans, what we see get lower with advancing age relative to young adults. In mice, we leverage a chemical or pharmacological approach to promote nitric oxide release. And there's a variety of ways to do this, but the method we use is excised carotid arteries that we then can't tie to fine tipped cannula that are in a pressurized system and submerged in a physiological saline solution. And we expose different drug compounds like acetylcholine, which activates nitric oxide release from the endothelium, and we give it different doses, and we do a dose response curve. And we can use a variety of drug compounds then to interrogate the mechanisms mediating these responses. And here's a zoomed in visual of a monitor that's attached to our stereoscope where we do these. So this is a cannulation from Brandon Wynn, and he cannulated a mouse carotid artery. So we've now moved away from these fine glass tip pipettes to these stainless steel cannula, which are a little bit more, have better integrity and hold tight to the vessel a little bit better. But we use these fine micro sutures to tie the vessel to these cannula. And then this is a pressurized system. So then we would look at the middle region of this vessel under a microscope and then be able to visualize measure the level of vasodilation with select software. And like with arterial stiffness, we do see a relatively consistent, similar magnitude of reduction if you compare young and older adults. So this is flow-mediated dilation, which we just described. Relative to young and old mice, this is the maximum endothelium-dependent dilation with acetylcholine. So a similar assay that I just described, and we see a similar magnitude of difference between flow-mediated dilation and acetylcholine stimulated vasodilation in mouse arteries. So again, inferring to us that the mouse model may be a translational model of human vascular aging. Well, if you two common mechanisms of arterial aging that have been very well established are excessive oxidative stress production and chronic low-grade inflammation. And the excessive oxidative stress with aging largely comes from excessive superoxide production, which feeds forward onto inflammation. And chronic low-grade inflammation can also exacerbate oxidative stress. And this is important because superoxide can act directly with nitric oxide and scavenge nitric oxide to reduce its bioavailability. So these are considered classical hallmarks or pillars of vascular aging or arterial aging. Unfortunately, however, large-scale clinical trials that have used chronic antioxidant therapy or chronic anti-inflammatory therapy have largely failed, such that they didn't transduce much benefit to the vasculature or cardiovascular health in general. So although these mechanisms are at play, ways to target these mechanisms and improve arterial health with advancing age, there's still a lot to be learned and a lot left on the table. So the approach that we started to take about 6 years ago was to then ask, what are potential integrative molecular events that might be contributing to excessive oxidative stress and inflammation, or what are molecular events that occur as a result of excessive oxidative stress and inflammation. And maybe by targeting one of these mechanisms or a combination of them, we might be able to then have better therapeutic targets for improving arterial function with aging. So this is this concept of alternative mechanisms feeding into these well-established processes. And what we took to was the hallmarks of aging. So this was a paper that was originally published in Cell in 2013. And it was largely to leverage the geroscience hypothesis, which if you're not familiar, it's the idea that there are certain molecular and cellular level changes that occur with advancing age. And if we can show that those happen consistently over time, that these mechanisms can then serve as viable therapeutic targets to mitigate diseases of aging. So that was first brought about in 2013. And then the same group updated it and called it an expanding universe in 2023. But there are many of these processes have remained consistent, but some of which were added were such thing as gut dysbiosis, as the gut microbiome has emerged as a therapeutic target for age-related diseases over this decade. But this really excited us as we were taking on a line of investigation in this process called cellular senescence, which has remained consistent across both of these time points. And then in a review article in 2022 in the Journal of Cardiovascular Aging, we took an approach of trying to combine the hallmarks of aging with what we know about the classic hallmarks of vascular aging. So in this illustration, we appreciate that oxidative stress and inflammation are at play, but we propose this idea that it's interacting with the hallmarks of aging to ultimately influence arterial health. And like I said, we focused largely on the process of cellular senescence. And what is cellular senescence? And like I said, it's a common aging hallmark, and it's been studied in the context of many age-related diseases, including Alzheimer's disease and related dementias. And I'm showing this little search from PubMed. This is from a few weeks back, cellular senescence and aging, and just to appreciate how much this field has grown over the last few years, now that PubMed indexes things on an annual basis. And a lot of these upticks, this isn't necessarily a lecture on the history of cellular senescence, but it's interesting to look at the significant upticks in publications around the time of select discoveries in this field. So it's something that being in the field, you have to appreciate the shoulders that you're standing on. But what is cellular senescence? In its most basic definition, it's when a cell is exposed to a select stressor. And with aging, that can be one, or chemotherapy or other genotoxic stresses. And with aging, it's essentially that cells only have so many cell cycles that they can go through before they have what's called replicative exhaustion. But these cells do not die off, they become senescent. And what that is defined as is an arrest of the cell cycle, but the cells remain metabolically active and alive. And they actually become resistant to apoptosis, or resistant to being cleared from our normal endogenous cell recycle processes. And this is thought to be pathological in part because senescent cells have their own discrete secretory phenotype, which is termed the SASP, the senescence associated secretory phenotype. And I'm illustrating with a variety of shapes and colors because it is a milieu of a variety of cytokines, chemokines, growth factors that can be secreted and act on a neighboring cell in a paracrine fashion, act on its own self in an autocrine fashion, or actually be released into the peripheral circulation and act in an endocrine manner. So this is thought to be a pathological driver of age-related diseases, and also a strong contributor to excessive ROS and inflammation. So thinking back to, we try to understand these mechanistic events that might be regulating excessive oxidative stress and inflammation. So therefore, based off this, we thought cellular senescence may be a viable way to go. So prior to getting into this work, and I think this nicely captures the translational research continuum and how you can move back and forth between animals and human research. So my colleagues up in Boulder published this paper in the American Physiological Society's journal, American Journal of Physiology, Heart and Circulatory Physiology. And it was the first report that vascular cell senescence with aging may be related to age-related arterial dysfunction. So what they did was employ a technique that we utilize, and it's called an endovascular biopsy. So through a cannula in the antecubital vein, you can take advance these J wires and scrape endothelial cells from the inner wall of the blood vessel. It's not painful. I'm seeing a lot of cringing faces, but it's honestly not too bad. the pressure is just from the cannula and maybe some saline flushing it out. But what's neat about this is you can take these cells and put them on slides and perform immunofluorescence staining. So now you're able to directly ask what is a molecular event that's changing in endothelial cells in older adults relative to young adults. And in these same patients, they measured endothelial function by flow-mediated dilation. So now they have the ability to look at the relation between vascular cell senescence and endothelial function. And they quantified the proteins P16 and P21. Those are just canonical senescence markers. So these part of a key senescence signature. And what they showed was that P16 and P21 were indeed higher in older adults relative to young adults and inversely related to flow-mediated dilation of the brachial artery. So this was the first line of evidence that vascular senescence may be implicated in age-related arterial dysfunction. But as the saying goes, correlation is not causation. So we took a reverse translational approach then and utilized a mouse model to then ask, does cellular senescence directly contribute to age-related arterial dysfunction? And to do that, we collaborated with the late Dr. Judy Campisi, who's referred to as the godmother of senescence research. And she was a longtime professor at the Buck Institute for Research on Aging in Novato, California. And her colleagues developed a mouse model called the P16 3MR mouse model. And not going to spend, could be a whole lecture too on how this mouse model was made. But ultimately what they did was develop a model where you could readily clear senescent cells that were marked with P16. So again, remember back to that previous slide, P16 is one of our canonical or classic senescence markers. So if we clear P16 positive cells, that would be a way, in the setting of advanced age, that would be a way of asking how does cellular senescence directly contribute to age-related arterial dysfunction. So briefly, so I can orient you to some upcoming slides, what they did was they inserted a herpes simplex virus thymidine kinase on the endogenous P16 promoter in this mouse model. And this is only really important because this is a viral disease put on this promoter that matters. And what we can do is then give 5 consecutive days of intraperitoneal injection in mice of this antiviral drug ganciclovir to ultimately target this virus and thus promote the P16 positive cells to be cleared via apoptosis. So what we did was we took young and old P16 3MR mice. We initially, just to provide a sense of what sometimes it takes to do age-related mouse research, you get these breeders initially. You breed up your colony, but then you have to wait approximately 3 years to study your old mice because old mice are around 27 months of age to be around the human corollary age of 75 to 85. So we had to sit and just wait to do these studies, but we felt like it was well worth it. So we took young and old animals and then gave them a vehicle control or ganciclovir. So you'll be oriented to 4 groups so that it was young and old, and then they got GCV or the control. And then we studied both female and male weanlings. And importantly, we did not see any sex differences. So the data that I'm about to show you is the 2 sexes combined. We ultimately published this paper in the Journal of Hypertension. And we're excited to find out just a few weeks ago that I was awarded the Basic Science Paper of the Year in hypertension. So what we found here, so here's aortic pulse wave velocity. Pre-treatment, so this is prior to giving the vehicle or the clearance of senescent cells with ganciclovir. We saw the presumed age-related higher levels of aortic stiffness as measured by aortic pulse wave velocity. So this is the gray circles. And then in the vehicle-treated animals, we did not see a difference over time, which is indicated in the black circles. But if the animals that were cleared of senescent cells with ganciclovir all had consistently lower aortic pulse wave velocity, now closer back to young levels. So this is indicating to us that cellular senescence may be directly implicated in age-related arterial stiffening. And similarly, we found with our endothelial function assay, where we gave increasing doses of acetylcholine and could generate dose response curves. So a lower area under the curve would be indicative of impaired endothelial function. So the black circles are our old control animals. And importantly, the old animals that were cleared of senescent cells with ganciclovir had endothelial function similar to the young animals. So it's indicated to us that with this genetic model that cellular senescence may be a viable therapeutic target for improving vascular function in the context of aging. So then, but you can't genetically clear senescent cells in people unless the CRISPR technology advances quite quickly. But one of the ways that this has emerged now that cellular senescence has become a viable therapeutic target is this concept of senolytic therapy. So it's a senescence targeted therapy. And if you're interested, I would highly recommend this review in Nature Medicine. It's from the leaders of the field of bringing senolytics to the clinic, Dr. James Kirkland, in particular, who is a gerontologist. But this, I like showing this figure because in physiology, we try to never say anything is good or bad. It's only bad when it's in excess. Everything has a normal physiological role. Oxidative stress is a great example. So senescence is illustrated here in the red. And you can see even at the youngest age, there is the presence of cellular senescence. Cellular senescence is involved in embryonic development, wound healing, and it's also strongly an anti-oncogenic process. But there's a theoretical threshold here that they've illustrated, and that you can get above this theoretical threshold in the context of aging, overweight and obesity, or with cancer therapy. And the idea is that when senescent cells accumulate, they can drive the development of chronic diseases and disorders. So that if we give senolytic therapy, that it could bring senescent cells back to a young, healthy level. But the goal is to not fully clear them because they do serve a normal physiological process. And importantly, to make a senolytic drug, we need to understand what is inherent about a senescent cell. And senescent cells are resistant to apoptosis, and they do that in part by upregulating this pathway called the Bax BCL2 pathway. So a common senolytic drug that you can use as a tool just to understand if senolytic therapy may be a viable way to go is to target this BCL2 pathway. And you can do that with this inhibitor navitoclax, or it's also called ABT263. So if you inhibit this anti-apoptosis pathway, you can then allow the cell to undergo apoptosis and go through the normal endogenous cell recycle processes in the body. So then that's what we did is we then performed a follow-up study in old animals where we gave them a control or this ABT263 with the goal of just determining proof of principle efficacy for the use of senolytic therapy to improve arterial health with advancing age. So also another way to orient you to this, or I'd like to orient you to the way senolytic therapy is administered, because like I said, it serves a normal physiological process. And so the way we think about supplements and therapeutics is chronic administration. But what's emerged in this field is high dose, hit and run intermittent dosing. So in humans, it's actually 2 days on and then you're off the rest of the month, 2 days on, off the rest of the month, and you might repeat that for 3 or 4 cycles. So it is a high dose of drug administration, but only for a couple of days. So one of the advantages is it also reduces patient burden from having to take supplements on a daily basis. But in mice, to translate the human ages to mice, what we end up doing is a week on, 2 weeks off, and a week on. So these are daily oral gavage administration of this compound. And similar to the genetic-based approach, we found that the old animals that were treated with ABT263 had lower arterial stiffness relative to the old control animals, and they had higher vascular endothelial function. So similar to our genetic-based approach, we found that a pharmacological-based approach that targets senescence could also elicit similar effects. So then this informed us that cellular senescence and senolytics may be a viable, cellular senescence may be a viable therapeutic target, and senolytics may be a viable therapeutic approach to improve arterial health with advancing age. So ultimately, we have this therapeutic target that we think from the lens of vascular aging, maybe we can now adopt this to cancer survivors. So now back to this origin, we're going to now flip. We set the stage with aging. Now we're going to move into cancer. And then as you can appreciate from this figure, second to cardiovascular diseases, cancer is the largest risk, or the second primary cause of death worldwide. And we've become increasingly interested in this relation between cancer and cardiovascular disease, as has the field in general. So it's probably no surprise to individuals in this audience, but aging is the primary risk factor for the development of cancer. And here I've pulled data from the Social Security Administration. So if you look at age at diagnosis, and this is delay adjusted rate per 100,000 persons. So this is it's a cancer diagnosis. So as advancing aging, or as age increases, we see a greater prevalence of cancer. And if we go back to one of the slides initially, that we have an increasing number of older adults, percentage-wise and absolute number, you would think that's a lot of it, lot more individuals that were being faced, that society and the healthcare system is being faced with, that are being diagnosed with cancer, if these older adults are presumably getting more cancer. And then this data from the American Cancer Society nicely shows that assumed trend is that here's the annual number of new cancer cases steadily increasing. And this year, by year's end, we're expected to be at 2 million new cancer cases just in the United States. But paradoxically, if we look at the number of cancer survivors and then that are in blue and the projected number of cancer survivors, that's only increasing decade to decade. So now this is really exciting is because we might have more cancer burdens on a societal level, but we have a lot more cancer survivors. So I think what that would tell us is that our means of treating cancer have become highly effective, and cancer is no longer the common cancers aren't killing us as quickly. And why that is the first line defense for cancer treatment, chemotherapy. These chemotherapeutics have become much more effective at treating the cancer. And we chose to focus on anthracyclines when we got into this line of work because it's one of the most common genres of cancer therapeutics. And it's first line for many adult cancers. So you have, it's used in prostate cancer, it's used in lymphoma, it's used in breast cancer, it's used in leukemia. So those, just again, more common forms of cancer. And the most highly utilized anthracycline chemotherapeutic is a drug called doxorubicin, or we'll refer to it as Doxo for short. And just to paint a picture of why that is the most commonly administered chemotherapeutic is that if we plot 3 common cancers, breast cancer, lymphomas, leukemias, that are commonly treated with Doxo chemotherapy, if we look at 5-year survival rate, we see a very impressive anywhere from 50 to 90% 5-year survival. So that means this drug is highly effective at treating the cancer. But this is kind of now the hook of the presentation, is that if we look at the cumulative dose of doxorubicin received throughout chemotherapy treatment relative to the incidence of heart failure, we see with it about a 20% increase in the cumulative dose, or maybe even less, we see more than a doubling of incident heart failure. So although there might be survivorship, we have some significant back-end burden that we need to deal with. And that's what's led to this saying that today's cancer patients are tomorrow's cardiovascular patients. And just to further paint this picture, to convince you from some data, here's data from the Journal of Cancer Survivorship. So this is time elapsed since cessation of chemotherapy relative to the cumulative incidence of cardiovascular disease. And importantly, they adjusted for age and sex here, and they see almost a steady rise linearly in cardiovascular disease burden relative to the time of cessation since chemotherapy treatment. So what we're dealing with is a large survivorship population. The survivorship population is increasing, but now we have a population that's of increased CVD burden, and that could occur in young adults, but also if older adults who already are at higher risk of developing cardiovascular disease undergo cancer therapy, they may now have a sort of double hit to the cardiovascular system, if you will. So that's where this field of cardio-oncology has emerged, that with cancer, we give chemotherapeutics, and it leads to cardiovascular diseases. And this is a growing field for sure. But then recently, this idea of reverse cardio-oncology has also emerged, that if you have cardiovascular disease, it's actually been shown to be an independent risk factor for the development of a first cancer diagnosis, but also it's a risk factor for cancer recurrence. So you can appreciate how nasty of a cycle this can be. But if you think, where can we interrupt this vicious cycle? And it's to improve cardiovascular health. So we allow these patients to go through cancer therapy with these highly effective chemotherapeutics. But what if they don't have to live with a severe cardiovascular disease burden for the rest of their life, and they get to beat cancer, but they don't have to have cardiovascular disease as a significant burden for the remainder of their life? And then if we can mitigate cardiovascular disease, maybe we then mitigate the recurrence of cancer. So this is now framed around the context of vascular aging. Why do anthracyclines lead to cardiovascular disease? When we got into this field, many folks had looked at how do anthracycline chemotherapeutics affect the heart. But what we know about how arterial health is a precursor to overt cardiovascular diseases, we wanted to view this through the artery or vascular lens. But before I show some data around that concept, just want to introduce you to a little bit of the history of this field. So in 1973, there was the first report of heart failure in a doxorubicin chemotherapy treated patient. And then there's been large efforts put forth to try to mitigate this cardiovascular disease burden. And there's one currently FDA approved drug called dexrazazane for the treatment of chemotherapy-related side effects. Unfortunately, however, this drug has actually been outlawed in some countries because it's been shown to reduce chemotherapy efficacy. So if this is our first line approach for mitigating cardiovascular disease burden as a result of doxorubicin chemotherapy, I think the field is still wide open. So the approach that we took is maybe anthracyclines are promoting arterial dysfunction. And if they are, if we can mitigate arterial dysfunction, we would presumably be reducing cardiovascular disease risk. So then to do this, we just viewed it through the lens of arterial aging and looked at arterial stiffening and endothelial dysfunction as we did with aging. And just to provide a little bit of background relevance of like what's out there. There are a few clinical reports. So here's a nice meta-analysis that was published in 2020 showing that anti-cancer therapies promote arterial stiffness. And here's select evidence that arterial stiffness may predict subsequent cancer therapy-related cardiac dysfunction in breast cancer patients. And here's some select evidence showing that childhood lymphoblastic leukemia treatment with chemotherapy could promote endothelial dysfunction. But if you look to the clinical literature, there's clear small cohort observations that these patients have arterial dysfunction, but the mechanisms by which this occurs are largely or incompletely understood. So that's where we now can go back to the mouse model to try to understand what mechanisms might be at play and hopefully take what we know from our field of vascular aging and our work in that space. So again, looking at the classic hallmarks of arterial aging, these are the first things we wanted to go after. And the first thing we did was look at the role of inflammation in mitigating arterial stiffening with doxorubicin chemotherapy. And to do this, when I started my postdoc, I was fortunate to be awarded a T32 fellowship through the Anschutz medical campus's renal diseases and hypertension. And we ultimately went on to publish this paper in hypertension, showing that tumor necrosis factor alpha mediated inflammation was involved with doxorubicin chemotherapy induced arterial stiffening. And importantly, this was in young animals. So then we took an approach to look at the other side of this classic pathway, which is oxidative stress. And with aging, I didn't say this earlier, but mitochondria in the arteries are actually a very key source of excessive oxidative stress. So if you have not thought about it before, I was surprised when I first heard this, that the vasculature is actually very rich in mitochondria, and it's not just in muscle. And was fortunate to then be awarded an individual postdoc fellowship in F32 to go after this question. And then we ended up publishing this work in the Journal of American College of Cardiology and their sister cardio-oncology journal, which at the time was brand new journal. We took a chance and it has emerged as the top cardio-onc journal in the field. So it's exciting to have gotten in there early, and they actually no longer take preclinical research. So we snuck in. So that's kind of led me to the last 2 years during my research assistant professor time up at Boulder, where I was awarded a K99R00 Pathway to Independence Award to now study the role of cellular senescence in mediating large elastic artery stiffening and endothelial dysfunction induced by doxo. And the way we tried to do this was paint this picture of like what led us to studying cellular senescence in the context of chronological aging. And now that we've established a similar premise with doxorubicin chemotherapy in young animals, maybe we can follow that same premise and target cellular senescence in the context of doxo to improve arterial health. And this is particularly important because if you recall, when I showed why does a cell undergo cellular senescence? Well, one of those is chemotherapy or a genotoxic stress. And chemotherapies can largely suppress cancer growth by driving cellular senescence because, again, it's an arrest of the cell cycle. So it's the direct opposite of cancer. So what we did was employ that same mouse model, and we gave them either a vehicle or a sham and doxo, and then followed up with ganciclovir or a control. And we found that the animals that just received the vehicle after doxo had elevated aortic stiffness as measured by aortic pulse wave velocity. And the animals that were clear of senescent cells were prevented from this phenotype. And we also found similar results with endothelial function, that the animals were cleared of senescent cells had endothelial function similar to the young control animals. And then we took a pharmacological dissection approach where we can then use different drugs in this preparation. And we use the mitochondrial targeted antioxidant MitoQ to acutely scavenge any excessive mitochondria derived oxidative stress molecules. And because the vascular endothelium is so responsive to excessive oxidative stress, by scavenging this MitoQ hours later in these vessels, we can measure restoration of endothelial function in this DOXO only group, showing again that mitochondrial ROS is implicated in DOXO-induced endothelial dysfunction. But importantly, the animals that were cleared of senescent cells had no further dilation in the presence of MitoQ, suggesting that if we can ameliorate senescent cells following doxo chemotherapy, we then can subsequently mitigate excessive mitochondrial ROS-related suppression of endothelial function. So now starting to paint a picture of a potential mechanistic pathway. And again, like with aging, this implied that cellular senescence may be a viable therapeutic target. We again employed the ABT263 senolytic approach using that 1 week on, 2 weeks off, 1 week on dosing paradigm. And similarly, we found that the animals treated with doxo but followed up with the vehicle had elevated aortic stiffness, but the animals that were given the senolytic were prevented from this phenotype. And we saw similar results with endothelial function as in the previous studies, and same thing with MitoQ. So now that this senolytic might also be suppressing senescent cells to then favorably modulate excessive mitochondrial oxidative stress. But during this time, we were also developing this line of work to ask, what is the role of the circulating milieu in age-related arterial dysfunction? And this is important because, as I mentioned earlier, the senescence-associated secretory phenotype can be released into circulation and act in an endocrine fashion. But the premise of doing this study was to build upon heterochronic parabiosis studies. So if you're not familiar with those, it's a very fascinating approach that's farly not translational, but it's where they sew two mouses' circulation together. So you can sew a young animal to an old animal, and the young animal takes on the old phenotypes, and the old animal takes on the young phenotypes. So you can appreciate we're not going to sew circulations together in the clinic, but what it does provide premise for is that there's a clear role of the circulation in driving some of these phenotypes that we're observing. So working with a very engineer-minded individual in my previous laboratory, we developed a model where we could, on a syringe pump, infuse serum overnight through the interlumen of a blood vessel. So we could expose a young mouse blood vessel to plasma or serum from an older adult or an old mouse, and then expose an old mouse vessel to blood from a young adult or a young mouse. And we found that we could flip the phenotype. So here's mouse serum in it. This is a young mouse vessel. So old serum could cause worse endothelial function relative to the young serum. And with the human serum, we could find the same thing. And like I said, this is important for us because the SASP can be released into circulation. So now if we see these phenotypes, it provides clear premise then to ask, well, what in the circulation is transducing these effects? But if without having this initial observation, you don't necessarily have full premise to go after that question. So this is an exciting line of research that also sets the stage for grants that I put in this past fall. And so that leads me now to current time, where we're engaged in the R00 portion of this pathway to, well, now it's the independent side of that award. And this title, Translational Studies of Cellular Senescence as a Regulator of Doxorubicin-Mediated Arterial Dysfunction. So now what we're taking is our animal work, and I'm working with two clinicians here on campus, and we're calling this the SENDOC study, Cellular Senescence, Doxorubicin Chemotherapy, and Cardiovascular Health. And I'm working with the Director of Cardio-Oncology, Dr. Lavanya Kondapalli, and the Director of Lymphoma Services, Dr. Manali Kamdar, to recruit adults that have undergone that are now cancer survivors, and they have undergone chemotherapy with doxorubicin-containing chemotherapy. And we're also recruiting age and sex match controls to ask, is arterial dysfunction present in these cancer survivors? And what is the cellular senescence burden in these patients? To provide then premise to potentially target cellular senescence to mitigate arterial dysfunction in these patients. And the outcomes in our study, we were initially funded to look at endothelial function and arterial stiffness, two methods that I've described earlier. We've also proposed to look at arterial stiffness through an approach called carotid artery compliance with ultrasound, and then been fortunate to receive a CCTSI CTRC microgrant to add additional measures to this study where we're leveraging cerebrovascular measures. And this we believe has some relevance because during our time when we were studying doxorubicin, we published a paper in experimental gerontology, which demonstrated that young animals that were treated with doxorubicin, when performing a memory assay, they had memory similar to older animals. And then if we compared the hippocampal transcriptome, so the memory center of the brain, to that of the transcriptome of the hippocampus of an old animal, it was quite similar. There was a lot of gene overlap. So this allowed us to establish that there's an accelerated aging effect on the brain transcriptome by doxorubicin. So now we want to look at cerebrovascular health to understand and cognitive health. So we'll do some cognitive function testing in these individuals. We'll also look at cardiac function through cardiac echo, and then also perform grip strength, as we do have some evidence from our aging studies that cellular senescence is implicated in changes in muscle function. And we also are allowing patients to have an optional endovascular biopsy where we could then probe senescence markers in these individuals. And we're excited about this because most folks have either focused on the heart or they have focused on the vasculature. So my grand hope for this study is that it will to date be the most comprehensive cardiovascular study. So where we're combining cardiac and vascular in this patient population. Then we're going to do a few studies with blood, the human blood, where we'll expose their serum to endothelial cells in culture and look at nitric oxide and mitochondrial ROS. We can expose the blood to young, healthy mouse aorta rings and perform an ex vivo measure of aortic stiffness. And then we also plan to use mass spectrometry or a variety of omics-based approaches to understand what in the circulation may be responsible for transducing these phenotypes. And then also as another biological tissue to quantify senescent cell burden, peripheral blood T cells have emerged. So we'll be quantifying senescence burden there. Then this is just a quick snapshot of preclinical future directions. We want to start studying this in the background of aging. We haven't yet to date because in our mouse model, we do see typical age-related attrition. So if you want to study 10 animals, you buy 30. So that's just the way it goes. So if we're going to give older animals also a drug that causes attrition, there's just like a lot of budgetary concerns that we why we haven't done this in the past, but it's obviously a prudent next research direction given how burdensome cancer is in the context of aging. And we're also interested in, we have some active collaborations to ask, how does cancer directly affect cardiovascular disease burden? So this is independent of chemotherapy. And we're also getting into different interventions, like lifestyle interventions, like treadmill running, or the combination of senolytics with treadmill running. We've started to employ a cancer-bearing animal model, and also looking at the effects of radiation, another common cancer treatment. So there's some ongoing projects in the lab. We were fortunate that Brandon Wynn was recently awarded the division's Integrated Physiology of Aging T32 project, where he is asking the question, does high-intensity interval training prevent doxo-induced arterial dysfunction, and does it do so by suppressing cellular senescence? We were fortunate to recently be awarded a CU Cancer Center Molecular and Cellular Oncology pilot grant, where similarly we're using high-intensity interval training, but this time it's in a breast cancer-bearing animal, and we're asking, does exercise training help deliver chemotherapy better to the tumor and less to off-target tissues? Does it enhance tumor vascularization? And then does it ultimately prevent vascular dysfunction? And some where we might be headed if NIH likes what we've proposed, we've targeted this notice of special interest research projects to develop oncoaging models. And so this was a co-sponsored NOC by NCI and NIA. That was submitted this fall, reviewed in February. And we also targeted this RFA by NIA, the impact of stressors on the biological mechanisms of aging and other aging-associated outcomes in experimental model systems. And then one thing that we're actively developing now is targeting this notice of special interest, advancing diet and physical activity biomarkers for assessing lifestyle interventions in cancer prevention and cancer interception research. And this is really to recruit patients, women with breasts that are at very high risk for breast cancer, but have not yet developed cancer. So the idea is to develop an exercise related approach. Well, that's what we're targeting rather than diet, to mitigate the increased risk in this patient population. And we're working with breast cancer oncologist on that project. And then because although we have talked a lot about more basic science, I think something that's been fun for Brandon and I, and with Carrie's mentorship, we've actually utilized Trinetics and Health Data Compass to access through the UC Health system a population of breast cancer survivors. And we looked at premenopausal controls, postmenopausal controls, and then premenopausal and postmenopausal women that had undergone breast cancer chemotherapy treatment. And we just did a broad assessment of typical cardiovascular disease risk factors. And that was recently submitted for American Physiological Society's annual meeting. So this is the abstract that we put in. With that, I think there was a few minutes for questions. I couldn't have done any of this without the support of our wonderful colleagues up in Boulder that I had for the 6 years preceding my arrival to this campus. I definitely have to thank the Division of Geriatric Medicine and the CU Cancer Center for their support. Obviously, the funding institutions here, the NIH, as well as the Ludeman Family Center for Women's Health Research, as we have an exciting line of research that's funded through them. And then definitely like to thank Brandon Wynn for coming to join me here as a postdoc and congratulate him on his recent T32. So with that, I'll take any questions. Thanks.
Excellent. We'll start with Josh and then go to Josh.
Hey, this may be an This may be an unrelated question or it was sort of tangentially related, but when you're talking about anti-synesthetic drugs, that sounds like we're trying to target a certain population of cells among another population of cells, which actually sounds like cancer chemotherapy as well. So these sort of as an avenue for therapeutics, what has potential in terms of drugs?
Yeah, so the NCI met, or leaders in the field of cancer and aging research met in 2019, and they published this think tank statement, is what they called it, in the journal that's put out by the National Cancer Institute. And the idea was that let the cancer drugs do their job. However, let's appreciate the cellular burden that occurs into survivorship, which is it's transducing a senescence effect on the cancer cells. But unfortunately, because the chemotherapy is not specific to the cancer cell and it goes everywhere, now we have senescence burden across our entire body. And so that concept that they advanced in this think tank statement was that into cancer survivorship might be a time when we can then leverage senolytic therapy to hopefully mitigate the subsequent pathophysiological events that happen into cancer survivorship.
Thanks, Zach. That was a really comprehensive overview of a huge amount of work in a field in a relatively short period of time. So big kudos on that. Kids get cancer. And sometimes they get doxorubicin. But cardiovascular disease burden is very low, typically in children. Do children treated with doxo have premature CVD risk?
Yeah, I have some of those slides and backup slides. So there is an ongoing longitudinal study at St. Jude's Children's Hospital where they're actually doing similar to the Framingham Heart Study where they're tracking people longitudinally, and the kids do indeed get elevated cardiovascular disease risk. But what you see in young is that there is a stress resistant phenotype. So it's not as consistent as it would be in like a middle-aged older adult getting it. But unfortunately, however, kiddos do get cardiovascular disease risks, and they've done nice comparisons against their siblings that did not get cancer and even twins. So now you've removed this genetic component of asking, is there a genetic background that might protect against? So I follow that St. Jude's Children's Hospital study quite closely to try to understand that, because that is where we'd also like to go with this work, is to the Children's Hospital and really hopefully give these kids a better life and not have an early death sentence because of cancer survivorship.
Thanks. I have a quick follow-up too.
It could, and that field is definitely emerging, and it's much more nuanced than I described. Is that you could have a P16 positive cell, and like the way that field has emerged to date has said, oh, that's a senescent cell. But now what we're starting to see is that there's P16 healthy cells, there's P16 high cells, there's mutated P16 cells. So now they're trying, these people that are making senolytics, this is where the drug discovery, drug development space is going, is to make senolytics that target senescent cells that have select mutations to get at exactly your point, because we don't want to drive cancer.
Hey, Zach, great job.
Thanks.
So a lot of these chemo agents throw women into menopause prematurely and maybe even throw men into andropause. How do you decipher the effects of ovarian or gonadal aging accelerated from those of just the cancer toxicity?
Yeah, it's a great question, and what these ongoing conversations that we've had in this space is what promoted us to look, or what prompted us to look in the Trinetics database. And interestingly, we saw, if we compared healthy, so non-cancer treated individual premenopausal women versus postmenopausal women, we saw the expected elevated CVD risk in postmenopausal women. But what was unique to our findings in the abstract is there were select cardiovascular disease risk factors like LDL cholesterol and systolic blood pressure that were elevated in premenopausal women that had undergone breast cancer chemotherapy treatment relative to premenopausal women that had not. So that would suggest to us that if estrogen is on board, there might be a suppressing effect on estrogen, because we know estrogen is cardioprotective. So our thought there is to maybe use this clinical evidence to go back to the mouse model to directly interrogate what is the role of gonadal sex hormones, both women and in men, to understand the direct role of sex hormone modulation by these chemo drugs. Because right now, to date, we cannot entangle that. So I think, yeah, we can use this clinical database to hopefully inform mouse research in the future.
Thanks so much, Zach. This is all so fascinating. And I have 6 questions, but I'll limit it to 1.
Okay.
So how do you mention measuring the burden of senescent cells? How do you measure that?
Yeah, it's senescence, unfortunately, does not have one marker. So we have to use a signature. We call it a senescent cell signature. And so we can do that in peripheral blood T cells by gene expression. We can measure common gene signatures. We can do it in the biopsied endothelial cells. And then we can also measure these SASP factors in the blood by proteomics. So we plan to take all of those approaches.
Truly impressive. Love all of the different methods that you are developing expertise and also advancing new methods, which is very exciting, which will have many applications. So thank you so much for this great presentation.
Thank you all.